Good. Good.
Yeah.
All right. Here's your mic.
Yeah.
Make sure that works.
Yeah, I'm gonna be wearing this.
Okay, yeah, that sounds all right.
All right. Hello, everyone. We'll go ahead and get started here. Thank you for joining us for Wildy Call this week. My name is Alex Schindler, and I'm a graduate student here at the University of Saskatchewan. And along with Anna, Kayla, Ilsa, Mercy, and Hannah, I help coordinate these seminars. So as I begin, I would like to acknowledge that we here in Saskatoon are on Treaty 6 territory in the homeland of the Métis. We pay our respect to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. So before we start, just a few housekeeping things. I would like to ask all of us joining online to please check and make sure that you are muted. And just so everyone is aware, this presentation will be recorded and later uploaded to our YouTube channel. At the end of the presentation, we will invite the audience to share comments or questions either through the chat box or by unmuting yourself. So if you have any questions about the webinar series as well and would like to be added to our mailing list, if you're not already, you can put that in the chat as well. So today is my pleasure to welcome our speaker, Dr. Mitch Weegman. Mitch started his career as an undergrad at Mississippi State University and then did his PhD at the University of Exeter in England, where he studied the demography of Greenland white-fronted geese. Mitch was a professor at the University of Missouri, where I actually first started in his lab, but about, 2 years ago, moved to Saskatoon with he moved with his wife, Caitlin, and now 2.5-year-old Elna. And Mitch is now a faculty member in the biology department here, and he is the Ducks Unlimited Canada Endowed Chair for Wetland and Waterfowl Conservation. If you don't know Mitch, he's an extremely positive and enthusiastic person, is super motivated by research and student mentorship. And so it's exciting for him to get to share some of the research that's been going on in our lab, which I am very fortunate to be a part of. So with that, help me welcome our speaker, Dr. Mitch Weegman.
Thanks, Alex. Hello, everyone. Can you hear me okay? Yeah? Okay, great. You're half deaf and you can hear me, Steve? Geez, it's good to see you, Steve. How long has it been? 7 or 8 years.
I think the last time we saw each other was when I was complaining when they were doing those bird counts.
Oh, yeah.
Bird counts.
The 4 square mile counts. Yeah. In the field with Steve and Bob Clark. You want to admit? Yeah, one more there. I can be watching that. I guess people will be coming up. Okay, thanks for the warm introduction. This is so cool. I was just saying I'm a wannabe attendee of this Wildy Call seminar series. So I'm openly saying I'm going to be at these things. I love these seminars. This is either my second or third one. When I was a postdoc with Bob Clark at U of S in 2015, I gave 1 or 2 of these. I can't remember, maybe departmental. So I'm really glad to see they're still going, and it's a treat to be here today with everyone.
So are you taking us all to amigos afterwards too?
We're going to philosophy, right, Alec? Yeah, we're going for a beer. You want to go?
Yeah.
Oh yeah, that's perfect. Can anybody know how to get rid of the bar on the top?
Or duck egg.
Floating meeting controls? Thank you. That's perfect. Okay, cool. So I thought today I would begin by talking about the Endowed Chair program. I understand from Ilsa and Alec, part of the motivation of having faculty speak at Wildy Call is to give you broader scope on our research programs than, say, one graduate student project. And so what I wanted to do to begin is talk about why the Ducks Unlimited Canada Endowed Chair is here at U of S, how it came to be, and then I'll talk a little bit about several projects we're working on in the group. So in 2017, a number of faculty around North America published a report called Who Will Mind the Marsh? And they detailed waterfowl-oriented faculty around North America. Here are all those folks. There were several endowed chairs in 2017 around the continent, all in the US, they're in red. And then there were some additional faculty in turquoise and in green, focusing on waterfowl. But the purpose of the article was that waterfowl and wetlands programs around the continent are in decline and have been for decades. And it was concerning. The idea that we may not have the base of training for future conservation professionals. So they prioritized where to invest in subsequent endowed chair positions around North America. The U of S, with Ray and Bob Clark, Ray Alasauskas and Bob Clark retiring, was prioritized as the lead place in North America. If you could invest in endowed chair anywhere, where would it be? The University of Saskatchewan. And so, moving forward, the next question was, how do we do this? There's a group of waterfowl folks, a group of conservation professionals saying we want an endowed chair at the University of Saskatchewan. Ducks Unlimited Canada partnered with the university to do this. This was a natural relationship. It's one that had spanned many decades. These two, if you don't know these two folks, these are two legends who are affiliated with biology for decades. That's Bob Clark on the left and Ray Alasauskas on the right. Greater than 20 current or former, that I know of anyway, DUC employees were trained by one of these two guys here at U of S. So this is a major training bed of wetland and waterfowl professionals, and more broadly, ecologists for the country. And so Ducks Unlimited recognized that they are a science-based organization, and so they invested in this endowed chair at the university. There's Ryan. Hi, Ryan. Welcome. We're just getting started. So Ducks Unlimited Canada invested $5 million, or started this $5 million campaign to endow a position at the University of Saskatchewan. At the time, the CEO, Carla Gwynn on the left, and president of the board, Jim Couch, were influential in doing this, saying we're going to act on this request by conservation professionals around the continent. And so they started this huge campaign. The initial agreement from Ducks Unlimited Canada was $2.3 million. This happened in 2019 to the U of S to say, we want to start this base and we're going to look to hit 5 million in the coming years. So the initial agreement we're under is 10 years. We've been raising money since this point. And now we're at 3.8 million. So we're getting there. And this has been a huge part of what I do with a team, a foundations team at the university and at Ducks Unlimited Canada, working on assembling the remaining funds to hit 5 million. We have a challenge grant at 4 million from Jim Kennedy, who's a philanthropist of lots of conservation activities around North America. And he and his foundation, Cox Foundations, have said, when you hit 4 million, we'll kick in the last 5 million. The last 1 million, I should say, to hit 5. So we're close. $200,000 to go. And it's within sight. And I can imagine over the course of this year, 2023, we'll hit that 4 and hopefully access the last million, and we'll have a fully endowed waterfowl chair at the U of S. So this is a really neat deal. The coolest part of this is that for perpetuity, depending on the global markets and investments, this endowment should draw about $40,000 to $60,000 a year for activities of the chair. These can be undergraduate research fellowships, graduate students, postdocs, field equipment, whatever is needed in our group at the time. So to have a predictable funding source like this to invest in important research activities around the country on how climate change and land use change are influencing wetland and waterfowl ecology, that's what this is all about. And providing these long-standing training opportunities to ensure that professionals are well situated to be able to tackle these global challenges. So I'll just end this first bit on the endowed chair by saying, sometimes folks think an endowed chair is one position or a university endeavor, perhaps. But what I want to say here is that actually this is a team of people all working to make this thing happen. So these are daily, weekly, monthly, annual activities, teams of people at Ducks Unlimited Canada and at the U of S who set this all up and are actively working to make this thing possible so that people like Alec and Ilsa and Lindsay Carlson and the rest of our lab can do what they do and be successful. So it's a super cool endeavor. They're rare, but I'm hoping they'll be less rare moving forward as we begin showing how these successes can propagate over decades, hoping that more of these come about across North America. So that's the Ducks Unlimited Canada chair. I wanted to describe it as well because I don't think it's common, at least when I was a graduate student, I had no idea about the different faculty of a university. All I knew is, okay, there's my advisor and I have a committee and maybe I take some graduate courses and that's that. But really the university at the graduate level, I think graduate students and postdocs should begin to be thinking about it more of, okay, you got research faculty, teaching faculty, you got research chairs, all these different appointments coming together to influence your all's training and the undergraduate training. And that's a really valuable thing when you're thinking about next steps, whether you want to continue in university systems or enter, say, the conservation community in another capacity. So now to overview my group. So broadly, we're tackling climatic and land use change through individual and population processes in birds primarily to inform their conservation. We're working really on two scales, the individual scale and the population scale. It's common for folks to look at either scale, but often not both. And yet I'm super enthused about both. And so we're asking a lot of questions, mixing these ideas. At the individual level, we're really interested in how movements and behavior explain individual reproductive success and survival. But actually before that, we're really interested in how weather and habitat explain movements and behavior with cascading influence on survival and reproductive success. So linking these things in a series of hypothesis tests has been really interesting ecologically. Also, at the population level, Dan Gibson gave one of these in November on some pintail population dynamics. A really good example, I think, of the questions about mechanisms for population change, whether survival or reproductive success more explains variation in population growth rate. And you can imagine among lots of populations, you've got some pretty big scale drivers, climate and land use change. We often work with migratory birds, so we're talking often about continent scale dynamics. Some understanding of population and individual processes independently allows a lot of sharing of information. And so you get this really cool feedback loop from individuals to populations and back again. So your power of inference is a lot stronger for knowing a little bit about both. And then with some information about the mechanisms, the movements, and the drivers of these scales, you can conduct scenario playing to say, okay, we anticipate the climate might change a particular way, or we anticipate, say, wetland drainage in Saskatchewan might change a particular way. And so if we adjust those variables to a reasonable amount of expected change, how might a particular, say, duck population respond? And what if we could alter the course of that change? Could you notice a change in a particular targeted animal population? These are the kinds of things you can do by understanding multiple scales. So that may seem a little bit of a black box. Don't know what that is. Okay, I get the individual and the population thing. I get the hypothesis testing. That all sounds good. Well, let's put some faces to some of these questions. Here are some of the species we're working on around North America, Europe, and a bit in Asia. So these shorebirds on the top are across a life history gradient, migration strategy gradient. These Hudsonian godwits winter in South America and breed in the Canadian and Alaskan Arctic. Long distance migrants, medium distance migrants, black-bellied plover. They're from sort of Central America, the southern US to the Canadian Arctic. And then avocets are short distance migrants. They're sort of Texas to Saskatchewan. These are Greenland white-fronted geese, the birds that Alec is studying. We're asking lots of questions about those birds. And then black ducks That's Elsa. Eastern mallards, new student coming on, Cassidy Weldrip, this September. At the population level, here are the suite of species. They're mostly waterfowl, also some other wetland dependent species, snowy plover in the top center there, and aerial insectivores, tree swallows there in the upper right. So lots of questions across quite a gradient here of migration strategies and short to long-lived birds. Okay, so what I'd like to do now for the rest of the seminar is split up the individual and population processes with a couple examples. I'm going to start with individual processes and talk about white-fronts, white-fronted geese, and then I'll talk about population processes and pintails. So we've been really fortunate in the group when I was based in Missouri and also here at U of S to have a suite of graduate students studying greater white-fronted geese. They're interesting for lots of reasons. I'm going to detail some of them now. We had a couple primary questions. A number of these students have been focused on a couple common questions of white-fronts at the individual level. One of them is, at what point are decisions about behavioral time allocation different between successful breeding and unsuccessful breeding birds, or those that differ? There are some individual choices here to disentangle. Also, we're interested in how do weather conditions mediate behavior and mask the effects of variation in preparation for breeding. We want to know, did you intend to do that, or were you influenced by the weather? And that's a really interesting question, because you've got individuals migrating thousands of kilometers, potentially under quite variable strategies, either because they planned on that or because they had some pretty darn serious weather conditions, blizzards, ice, adverse wind directions. Lots of things can cause birds to go astray or to not be able to live out the decisions that they were intending. So if you can disentangle that, okay, it's getting really interesting. To study these white-fronted geese, have you ever seen these neck collars before with Alex's work or any? Okay, so this is how we study these birds at the individual level. We put a neck collar tracking device on them. There are two solar panels at either side of the neck collar, and these collars last a year or two, sometimes more. They collect the GPS position every 15 minutes and an ACC or acceleration fix every 6 minutes. The interval is a little variable depending on which population we're studying. The data are uploaded via the cellular phone network, global cellular phone network. So we basically buy a cell phone plan for each bird. When they're in cell phone coverage, they upload their data. And typically we set this interval to daily. The acceleration data allow us to get at behavior, something that has been quite challenging to get at other than more classical observations of time activity budgets. So this stuff we're collecting every 6 minutes, the ACC data I'm referencing. Initially, the acceleration data are recorded in 3 axes. So front, back, side, side, up, down, motion. And we don't really know the motion or the patterns associated with particular behaviors. So we put this tracking device on a captive bird, or we go visit wild tag birds, and we film them for tens of hours, 30, 40 hours. And we form a library of truth. So we know exactly the time on the tracking device. We know the behavior of the bird through the film, and you can link this stuff and say, okay, that pattern, didn't know what that was previously, that's feeding. And the stationary, well, that's a little bit easier to work out. That's a flat line, 3 flat lines. And then the flight is something we don't often observe, but because these are migratory birds, they fly for tens of hours during migration, and it's the only time you see that amplitude and frequency that year. And so we pull fixes of flight. And in this way, you can train machine learning algorithms. Here are a variety of them, neural networks and regression trees and random forests. And you can do some really neat subsampling, or you can pull truth out of this model. And then you can test the model on itself and say, how accurate are you with missing information? And then, oh, well, actually, we have the missing information. And so you can work out some level of accuracy, a mean and a standard deviation. And then we can select the one with the greatest accuracy, or the one that has this mixture of mean and standard deviation that is acceptable. Once you've selected a machine learning algorithm, you turn the thing loose on millions of fixes for which you have no idea what the behavior is, and it classifies them all. It's pretty darn amazing. And we do this for millions of fixes for hundreds or thousands of migratory birds at once. Stephanie Cunningham was a master's student, my first graduate student actually at Mizzou. She started in early 2017, finished in 2019, and she was interested, or collectively we were interested in trying to get at reproductive status with these GPS acceleration devices. These birds nest in the far north, in the Arctic, around the world. Very difficult places to get to. You can invest a lot of money and not actually observe a whole lot of nesting. So we were using these devices to get at productivity, reproductive success, because it's believed to be the mechanism for population change. Survival of Arctic geese is often high and quite stable. Productivity can be really variable. We don't know why, because we can't get to the far north regularly. So we're using devices to work out retrospectively what reproductive success likely is. Here we're using energy expenditure. This is a metric that you can get from the acceleration data. So you can convert ACC data to overall dynamic body acceleration, which is a proxy for energy expenditure due to movement. And you'd expect that a nesting bird, an incubating female, would be relatively stationary for a particular set of days corresponding to the incubation interval that we anticipate. And so what we're looking for is a big drop in the reproductive window to ascribe full-term incubation to an individual. We have lots of individuals for which that drop never happens. And so we assume that that individual went to West Greenland, had a wonderful summer with the muskox and the caribou, never nested, and then flew back to staging in wintering areas. So when you plot this stuff, it becomes pretty apparent who attempted and who deferred. So we have several questions about these Greenland white-fronted geese. One was, does time feeding prior to the breeding season predict decisions to defer or attempt reproduction? For this, we relied on an ecological memory framework. So here we're getting at the idea of past states influencing present or future responses. Here's an example of what I mean by that. So here on the x-axis, we have day of migration from winter to breeding areas. On the y, we have the proportion of time feeding and some weights. So this black line is a made up proportion of time feeding line, hugely variable depending on the stage of migration. The purple bars are weights. This is the cumulative importance of each day. So these ecological memory models allow us to assess relationships day by day, the relative importance of day one on breeding success, day two, day three, but also they allow us to get at this weight. Do a sequence of days become more important cumulatively toward variation in reproductive success? So here's a predicted relationship where day of migration isn't super important cumulatively. As the birds get closer to the reproductive period, we know their physiology is changing rapidly. We assume that, holy cow, if you're planning on breeding, you better start feeding because you're going to get to the Arctic and there's not going to be a whole lot to eat. And yet you got to nest in a particular interval because those goslings have to fledge before first frost so that they can get out of there and get to Iceland. So this is what we anticipated seeing from this kind of analysis. And I'll detail a couple more predictions. So related to the amount of time feeding, we expected that attempting geese will show increased feeding behavior compared to deferring reproduction. And here's what we saw. So on the x-axis there, we have energy expenditure. On the y, the probability of breeding deferral Steph was looking at. And so we see here that as energy expenditure increased, the probability of breeding deferral also increased. We did the same thing for proportion of time feeding. And we saw here that as proportion of time feeding increased, the probability of breeding deferral decreased. So a couple things that you might anticipate among birds intent on deferring or having a go. I should have said earlier, these geese, they're long-lived. They have this luxury of being able to decide annually whether they breed or not. In shorter-lived animals, say even ducks shorebirds, and smaller songbirds say, they have to have a go every year. They don't live long enough to do anything else. The geese can arrive at the Arctic, all intent to do it. We're going to go, get there, lots of snow and ice. Nope, not this year. So that's what we're trying to get at here is what's up with deferral and reproductive success? Is there a relationship? And we're seeing some neat indicators with energy expenditure and proportionate time feeding. Okay, I mentioned a really cool aspect of the ecological memory models is weight, daily weight. This is the purple histogram I had a second ago. That was made up previously. Here's the actual estimates. So we don't see any change in the estimates of daily weight. And I'm not going to dive into a ton of details on this, but we're seeing this same pattern. Initially, we thought, oh my gosh, each day is as equal as any other day. Holy cow. Like what you do in winter, this is the x-axis, winter day 0, breeding areas day 50. What you do in Ireland matters just as much as what you do right before the reproductive period, which is crazy. That is not commonly predicted. And so we thought, oh, geez, that's really cool stuff. That's really interesting. Well, then this is with Stephanie. Alex explored it quite a bit more. Holy cow, Ilse explored a little bit, and we're starting to see some patterns that among species, this is common. So it's led to either a genuine, wow, that's different ecologically than what others have found, or there's something about this framework that we're not able to properly capture the ecological relationships when sorting time to day. So there's got to be something going on here. It's a series of simulations for a motivated student, probably one of these future graduate students to explore and disentangle. It's still a very interesting question to me. Okay, the second question with Stephanie's work was, do weather effects on feeding behavior change within the migration period? And for this, we use dynamic linear models. Really neat. These models allow time varying responses and covariates. So we have this really frequently collected behavioral information, acceleration data. We know weather is quite dynamic. Geez, it'd be nice to estimate effect sizes at the interval at which these things can be collected. And this modeling framework allows that. So we're really interested in this work in examining that temporal variation. A couple of predictions on proportionate time feeding. We thought that precipitation rate would be negatively related to proportionate time feeding, and minimum temperature would be positively related to proportionate time feeding. So this is getting at that idea of intent I was referencing. And here's a hypothesized relationship, effective precipitation on the Y and day of migration on the X. We thought that effect sizes would be greater later in the migration period. So these things become more pronounced as these birds are being squeezed about to lay eggs and begin incubation. Here's what we see. So this is a bit of a mess. Can you see the rectangles in the back there? Yeah? Okay. Let me break this down. Each row is an individual bird. These names are about females and males and what year we put the device on, and no big deal, but just know that each row is an individual goose. Each little rectangle is an individual day. And then these filled and open squares on the right are reproductive status, where filled was successful, full-term incubators, that is. And so from this, we see, oh my gosh, there's a ton of variation here. We were expecting uniform relationships. Really bad weather influences everybody similarly in migration, say, or in this case, on proportionate time feeding. So red would be the negative relationships and blue positive, and you'd be expecting that all blue, all red, the days, the period of migration is not the same among individuals, so you have more or less information about those. And yet we don't see that. We see hugely variable responses. And sorry, red is positive and blue is negative. I think I said it the other way around. But the point here in my mind is that you got a lot of heterogeneity, a ton of individual responses day by day to a particular set of conditions. We also modeled the relationship with temperature and repeated for energy expenditure. So we have 4 of these things. I've just shown one to give you an idea of the scale of the relationships. So these are effect sizes for each individual, each day of migration. Really, really cool stuff. So some conclusions here. Energy expenditure and proportionate time feeding explained attempt versus defer. And yet we need to quantify the food quality to understand these contributions more deeply. So far, we've treated the proportionate time feeding as if all foods are the same. But we know at each stage of migration, they're feeding on slightly different stuff. And even within a stage, they're feeding on different stuff. I have been pitching for a few years now this idea of going out in real time to those tagged locations, sampling the food that that bird was feeding on because the data are uploaded so regularly, and doing this at the continental scale, running around following individuals, big armies of technicians, and you're running all these samples. And whenever I describe this, people say, you're nuts, dude. Like, that's crazy. We're not going to fund that. And so I haven't been able to pull this off. But in the discussion sections of these papers, you can imagine this is a pretty darn important assumption. And so at some point, we're going to have to get at this among projects. We're going to have to understand and relate the level of nutrients exactly to the amount of time that these birds are feeding. So that's what's on the horizon in my mind. In terms of the second question, we showed, I think pretty convincingly, that precipitation and temperature can strongly influence behavior, but not uniformly in the way that we may have expected. So there's the potential to mask the effect of variation in preparation to attempt and defer. And it's something that we're exploring a whole lot more with Alex's work. So I just have a couple slides on Alex stuff. I don't want to steal, like he should do a whole Wild to Call seminar because there's a ton on his work on the full annual cycle. I've been relaying to you this business of following these birds from winter to breeding areas. So that's, say, March to May, June kind of thing. And yet there's a lot of examples. There are a lot of examples in ecology that the full annual cycle, particularly for migratory animals, is very important. And so that's what Alex's been looking at, is taking this a step further. When we looked at all those individual day-by-day relationships, the lack of importance in the weights, but also all that heterogeneity in the effect sizes of proportionate time feeding and say precipitation, we kind of took a step back. Here in my PhD 10, 15 years ago, we were looking at it from like 2-week blocks. There's Phil. Welcome, Phil. We were looking at this from a 2-week block period, and that just seemed too broad. These individuals are making decisions at finer scales than that. We were assuming from a human mind 14-day intervals for migration. So we went super narrow with Steph. We want all these individual relationships. And yet when you do that, well, holy cow, it's a hodgepodge of answers. So we backed out a little more with Alec, but to more reasonable, easily defendable ecological periods. And it's these 4. A late winter, that's a prelude to migration. The first half of staging, second half of staging. Both of these are in Iceland in the case of Alec's birds, the Greenland white-fronted geese, and then early breeding in West Greenland. Testing similar relationships, weather, extreme weather events on proportionate time feeding and energy expenditure. Here are some of those results. So we're seeing here the effect of days below freezing on spring proportionate time feeding, some pretty strong relationships for late winter and the second half of staging. And I have a few more plots from Alec here. So that was days below freezing. Here's the same thing, but time in grasslands. So grasslands are super important for Greenland white-fronted geese, dominant food. And we see this declining strength of effect size from late winter to early breeding. So birds getting maybe what they need earlier, and then the importance of that proportionate time feeding declining right prior to reproduction, just an early hypothesis. And then we're linking these extreme weather events and behaviors with breeding success in a hierarchical model. So Steph did those 2 analyses separately. The ecological memory model and the dynamic linear model. Alex merged these in one model with full air propagation to understand how these things are connected: weather, behavior, reproductive outcome. And so here's some early results from Alex's work showing the spring proportionate time feeding on breeding success. Looks like late winter and early breeding proportionate time feeding are most positively related to successful breeding. Full-term incubation. I mentioned Alex exploring the annual cycle. So that was all in the prelude to breeding. Here's in the post period. So this is the effective days below freezing on fall proportionate time feeding. So we want to understand now some fall effects as birds are leaving Greenland and they're going back to Ireland and Scotland. And here it looks like days below freezing, that relationship is strongest in early winter, not in that summer or fall staging period. Here we have the effects, again, of time in grasslands on fall proportionate time feeding. And we see this late breeding period, late summer and early winter, popping out with some negative relationships on staging areas. Just like in the previous slide, we can link the weather and proportionate time feeding in a bundle, in a hierarchical model, to some level of understanding about fitness. Except here we've turned the tide a little bit. We turned the table. And now we're asking about, is there a cost to reproductive success? So here we're asking, okay, if you bred successfully or you didn't, does that influence what your proportionate time feeding is like in the fall? The idea would be that mom and dad raising kids are able to refuel at a slower rate than those without. And we see some evidence of that. Also, we're trying to link these effects of proportionate time feeding on survival. And so here we have some weak evidence, I'd say, of proportionate time feeding on survival. You feed more, you live at a little bit of a higher rate. So it appears that there's a bit of a trade-off, but again, early days, I think, on interpreting these results. Alec will be writing this up pretty soon. Okay, that's mostly with Greenland white-fronted geese. I mentioned that we've had this amazing team of students on also mid-continent white-fronted geese in North America. A couple of years ago, we learned that there were 7 independent research teams studying these greater white-fronted geese around the world, all deploying these devices in real time. Totally nuts. Incredible revelation. This is the North Pole. Here's North America, there's Greenland, there's Europe, for a bit of a reference. So you're looking at the world from the top, and the different colors are the different populations of greater white-fronted geese. These are their migratory paths. And we've had a couple postdoctoral fellows in my group, Amy Shipley and then Lindsay Brodus, who recently started. And these folks are going to be comparing migration strategies of the world's white fronts. We think there's a lot of value in understanding a lot about one population and how that compares to others in terms of flexibility of migration, and even how they're resilient to landscape change. You can imagine these different migration strategies are crazy. You got birds flying through Beijing, flying through rural Saskatchewan, flying coastally in the case of Pacific flyway white-fronted geese, same genetic profile, totally different strategy. So there's a lot to uncover here and learn about ecologically from an analysis or series of analyses like this. Okay, so we are broadly now with individual processes quantifying these various features of movements, behavior, reproductive success for conservation utility. So we're discovering how behavioral time allocation and movements among habitats affect subsequent decisions to attempt or defer breeding in a wetland-dependent migratory species. And we think with this information, we can prioritize conservation investments. So where and when, wintering areas, staging areas, breeding areas, for maximum effectiveness in the face of global changes. So there's a lot of utility in this kind of work. Now I want to switch gears for a few minutes to talk about pintails. So we're going from the individual level to the population level. A few examples. We're testing hypotheses about how land use and water explain pintail population dynamics to inform conservation investments in the prairies and across North America. Here's a conceptual model that Dan highlighted of pintail demography. So these are migratory birds. They breed in the prairie region, in this northern unsurveyed region, and in Alaska. They winter in California and along the southern Gulf Coast. There were some, this is from Brady Matson in 2012. There were a series of hypotheses about how fall and spring migration might differ. Subsequent postdoc on Brady followed Eric Osnes, and he showed there was a lot of variation in pintail productivity dependent on where they bred. And so we've been exploring that for a series of years with a series of students. This is the prairie pothole region. We're in the heart of it. This is the most productive area for waterfowl in North America, one of the most productive in the world. And one of the primary reasons why all those waterfowl faculty of North America said we want an endowed chair at U of S. This is the place, the duck factory of our continent, and yet there's a lot of variation spatially in how these landscapes might be conserved, also politically, of course, by province and by country. And so we split these into different ecostrata. If you've flown over the prairies at relatively low altitude in summer, you might have this view. This is a relatively wet year, lots of potholes on the landscape, and yet lots of cropland. These pintails nest in cropland. They're drawn to shortgrass prairie. The surrogate shortgrass prairie of today is stubble. And so these birds are drawn to stubble, they nest in that, and then you have machines in spring planting and destroying nests. Agricultural practices across the prairies have changed big time. This thing called conservation tillage has increased hugely. We use this as a proxy for agricultural intensification. The idea in history that farmers would leave fields fallow for a year, every other year they would plant to minimize soil erosion, maximize soil nutrients. Today, in today's farming world, you can manage to plant every year. So this conservation tillage has increased a lot. Grassland, the whole grassland, native, but also hayland, has mostly stayed stable across these ecostrata. At the same time, I mentioned prairie pothole region full of wetlands, and yet the wetland dynamics in each of these ecostrata has been really variable over this same time series, 1960s to present. Sorry on that. We're trying to link wetlands and agricultural intensification with pintail productivity, reproductive success. We had a couple hypotheses about pintails. One is that as the number of ponds increase, the pintail population would increase. Also, that agricultural intensification was negatively related to the pintail population. And importantly, that the effect of agricultural intensification had sort of outmatched the number of ponds. So the negative effect of agricultural intensification is stronger than the positive effect of ponds. Also, we predicted that there was a stronger correlation between ponds and productivity under greater agricultural intensification. So under the most human changed landscapes, the importance of ponds becomes the most important. For this work, Xing Zhao, a former postdoc in my group, was leading it. We formed an integrated population model for pintails. These models combine three common data sets in animal ecology: population survey information, banding data for survival, and productivity via age ratios. Typically, people use this harvest survey of wings collected annually, but we were using age ratios at banding. Tens of thousands of pintails are banded across the prairies every year, juveniles and adults. That ratio gives you a bit of a spatial proxy for productivity annually. I'm not going to get into all the details of Xing's work and the integrated population model, but I do want to show you some of his main findings about the contributions to population growth rate. One of the main things demographers often want. Here we have productivity in blue, female survival in pink, and male survival in brown. And no matter which ecostratum you look at, we see that blue is a primary explainer. Productivity is a primary explainer of population growth rate. Related to our hypotheses on agriculture and ponds and on productivity. So here again, each plot is each ecostratum. We have an interaction here. So the lines are the views on agriculture. Blue is high agriculture, red is low agriculture, and then the x-axis for each of these is the number of ponds. From this, we see, as we anticipated, the blue lines, those slopes, are steeper than the red lines. So the influence of or the importance of ponds becomes more important under the greatest agricultural intensification. Y-axis for all this is proportion of juvenile females. So you're able to get the most proportion of juvenile females by having a ton of ponds. So agricultural intensification resulted in lower pintail productivity, high sensitivity of pintail productivity to ponds. It's our climate index for variation. And we think there's some pretty important implications for other grassland and farmland birds. Perhaps you heard of or read this paper, Ken Rosenberg and others published in 2019 in Science on the potential risk of species around the world and the loss of birds. It was a huge deal, 5 billion birds lost, made a lot of national and international news coverage. One of the species, or groups of species, I should say, doing quite well, the only one was wetland birds. But in the pintail case, we're in this grassland setup. These are nesting in grasslands, in farmlands. And so you can imagine that a lot of the grassland species are like pintails, suffering under agricultural intensification. Okay, I mentioned one of the really cool things we can do when we study individuals and populations is conduct scenarios. And so I want to show you some of the scenario playing we've been doing with pintails. There are 3 metrics we change with pintails: climate, reducing drainage, and reducing agricultural intensity. In this set of scenarios, we've just chosen a positive outcome or a negative outcome. Can you change it substantially or not, given predictions of how these things are really going to change in the next 10 and 20 years? There's quite a bit of work published on all 3 metrics, so we were pulling from the literature. So here, I want to show you, hang on. That one? Thank you. This is Alberta parkland, one of the ecostrata. And we did the full mixture here of combinations. Basically, what I'd like you to know is that all the red combinations are really bad. All the blue combinations are really good. Maybe you're not super interested in that, me either, necessarily. We're interested in the middle. Okay, you can't influence all 3 metrics maybe. What if from a habitat conservation perspective, you could influence one of them? Maybe you could change policy related to drainage or to agriculture. What might you anticipate a pintail population responding like? So we did that for all of these ecostrata. And the y-axis here, the x-axis are not the same, sorry. And so depending on which ecostratum you're interested in, we can begin to see, depending on which metrics we might actually be able to influence, where we invest dollars to begin a better, more informed conservation effort. So this isn't made up stuff. It looks like some simple levers. Well, it is some simple levers, but it's all based on how we anticipate these metrics might change in the future. And it begins to inform priorities, millions of dollars on the ground, which is pretty darn cool. So returning to this continent scale model, we want to know the prairies are really important, but how do the prairies fit in the continent scale? And so Xing formed an even broader continent scale model, linking these individual breeding regions, Alaska, northern, partially surveyed, and prairies. Again, looking at all the demographic components, survival and productivity. Here on the y-axis, we have total population growth. And even at the continent scale, we see productivity is the primary mechanism for population change. Okay, a couple more slides. Xing moved on, got an awesome quantitative ecologist role with Bird Conservatory of the Rockies. And now we have a new postdoc, Dan Gibson, who started in our group last summer. Dan has taken the skeletal continent scale model and really magnified it. He's done a phenomenal job adding in additional components of demography that we think are important, like harvest, for example. So in the previous example, we were looking at relatively simplistic views on demography to population growth rate. Now we're able to take a more advanced approach called life table response experiments, in which we can look at the variance of each of these demographic components and relate them more robustly to population growth rate. So here on the x-axis, we have multiple things going on. The first whole row here are females. The second whole row are males. Previously, we were looking just at females. Here we have fecundity. We have now seasonal survival, so winter survival. We have harvest, and we have summer survival. This whole batch is hatchier birds. These are juveniles. This whole batch are adults. So we can split this according to sex and age, and also the entire suite of demographic contributors. The general horizontal lines are sort of no effect, and then we can start to see some positive and negative relationships. I'm not going to detail all this. More, this is a quick hit to say, holy cow, we went from a skeletal continental model to something that's now really approximating, we believe, the ecology of these birds at a pretty big scale. We can go finer scale than that. Here, you're looking at females across the top, across the 3 breeding regions, males on the bottom across the 3 breeding regions. This is for every year now that the banding data, these demographic components are available, 1961 to present. So before we were looking at means, now we're looking at the relative contributions, again, of each of those demographic rates on the top. So each color is a different demographic component. Fecundity is gray, the one that Xing showed was quite important. And now we can see via these bars, the width of the bar, positive or negative relationship, begins to tell us, okay, in what years did each demographic component become more or less important? We can conduct a whole suite of hypothesis tests on this, magnifying that land use and climate change stuff I was just talking about. So we went from this holistic view on how the system was working to some really fine-grained views. And I think there's a lot of might in both of those scales to really holistically understand pintails and a number of other well-independent migratory bird species. So next, we're going to finalize hypothesis tests of the breeding area and wintering area predictions, how climate, land use, and also body condition might be explaining variation in demography. We also would like to conduct scenario playing similar to that prairie model I was showing you, but at the continent scale. If you can invest in wintering areas, breeding areas, or somewhere in between, where would you do that? How can you have the greatest impact on pintail population dynamics with limited financial resources? So we're about to do that. So we're discovering here how populations respond demographically to the direct and interacting effects of climate and land use change. And we can provide the ability to generate scenarios to inform different land use program and policy decisions with respect to those regional and continental climate change projections I was referencing. So we have this really neat capacity at the population level to better inform conservation. With that, I really appreciate the opportunity to talk with you all today and happy to take any questions.
Awesome. Thanks, Mitch. So I guess we could start if anyone has any questions in person.
Yeah, I don't really need this, but so my question is, you've broken up these populations into these kind of big land chunks. Do you know what the minimum resolution of space that you get variation is? So, if you had a model that had a greater spatial resolution, could you start to resolve these spots are where the good food is and these spots, could you account for some of these things that might be, maybe they're individual variation, but maybe they're also a finer spatial resolution?
Yeah, it's a great question. I think we could, but with slightly different data. So the data that are informing this model are, a mixture. So we have these banding sites. They're spread all over, hundreds of banding sites are spread all over the prairies. You could look at age ratio at banding, productivity at each banding site. And that could be some level of fine scale space, finer scale than what we were looking at. Now, because all that was an integrated model, the breeding population survey we were using was at the strata level. So the breeding population survey is a huge prairie pothole region wide endeavor, 30, 50 kilometer long survey. And so you'd have to take that scale and somehow compare to the super banding site scale. And so these things become a bit harder to disentangle, I think, when you're trying to keep the same reasonable apples to apples comparison. Now, separate to all this, I've been working on tagging pintails. And so Bart Ballard, a colleague and friend at Texas A&M, is leading this. We put 500 tracking devices on adult female pintails the last 3 years. So they are going to these super local scales, and they allow us to link populations on the survey with the age ratio at banding. So I think those tag birds are going to be critical for marrying these different scales that you're referencing. So can we do it? Yes, with additional data on scale. Can we do it with the data we have right now? Like, could you just simply splice it finer scale? I don't think so. So we're headed there as soon as we can include additional information.
And for the whether good food or bad food for the ones you do have the GPS data, can you do like these ones all were on a very similar, I'm not a bird biologist, so I have no idea what I'm talking about, but could you say like these birds all migrated in a very close spatial area? So we don't know whether it was good food or bad food, but it was all the same for them.
Yeah. So now you're on the white fronts, you mean, or those pintails I'm talking
The
about?
white fronts.
Yeah. So were you asking originally about pintails?
Both, yeah.
Both. Okay. So I was responding on pintail limitations, but now I'll switch to white fronts here, just so we're all on the same page. So on the food quality, definitely you can do that. Even in the absence of the nutrient crazy study that I was referencing, you can start to look at migration metrics, number of stopovers, amount of time in a stopover, lots of spatial layer, cool comparisons of why do all these individuals go there and some go over there. What about differential reproductive outcomes associated with those strategies? So we can link all these things holistically for the tag birds, no sweat, under the presumption though that that's representative of population scale dynamics. So the true, in my mind, all singing, all dancing is to do the individual level analyses and have the integrated population model for the same species. And then you can imagine both processes twirling in the same model. So you got individual effects cascading up to population level effects. Full error propagation would be nuts. And so we're going to have a team of postdocs working on that in the next couple of years because I'm just so excited about it.
Anything else from in-person audience?
So I have a question about the first part of your talk about the white fronts.
Sure.
When you were looking at the weight of importance of days, and you kind of found like, wow, they're all, every day is just as important as any other day. And you said that was starting to be discovered in a lot of other species or was known to be a fact of other species. Do you know if that's just for migratory species or if that's across the board has been found in like non-migratory species, or is this only in birds, or what other species has it been seen in?
Yeah, great question. So I was referencing only the work I've seen firsthand from our group. So those are all migratory birds. In general, this ecological memory framework, I mean, Alec, you know it better than me. I don't think it's been widely used yet in ecology. This work came out over the last 5 or 6, 7 years, and there's a lot of potential for it in my mind. But when you do this search on Google Scholar about who's using ecological memory and for what systems and what research questions, there's not a whole lot yet there. And so I don't know how common our problems are, to the point that we were at the moment of Alec literally recreating the couple seminal papers and trying to get at, can we observe what they observed and then convert it into the migratory bird world. They were looking at it with plants.
Yeah, there's a lot of like larger scale sort of weather and climate patterns, how it affected like net primary production, micro, what was originally developed.
So we haven't, in my mind, I haven't seen convincing evidence since then that this can be applied to the sorts of ecological questions that we're asking. We saw potential for it, and I still think there is, but there's this lingering question of how well suited are the timescales and the variation within and among individuals for that particular framework. And do we need to tweak that framework or develop a different one that's a little more flexible for time? Say the geese don't operate daily or even maybe every couple of weeks. The ecology on those birds might be they're purposefully set up or through evolutionary time set up to be able to handle lots of environmental variation, except the most extreme environmental variation. So you can imagine a system where most of the year, no need to measure daily effects or even biweekly effects. You can measure effects according to month or a couple months, except for the really important events during migration that really whack them. Those ones could be measured like by the day. And so but this system isn't yet flexible for that level of environment temporal variation. So that's where I'd like to go with these explorations is let's create a bunch of different environmental variation over different time scales. And let's see whether we can recover truth in a simulation framework.
So for that, that ecological memory modeling, I assume the question is, does individual variation mask this? And so, not really, is the individual variation like continuous, or do birds show like subpopulations? Like these are the ones who like to eat early in the year, and these are the ones who like to eat late in the year. Or is that?
I don't think we know yet. I think within the realm of possible, like given the particular life history of these birds, I think we're seeing a lot of different choice. And to me, that suggests that there aren't, for most parts of the year, there aren't massive pressures constraining all the same decision. So you can feed over there or over there, and both are going to be an okay outcome today and probably tomorrow too.
Yeah.
But there may be parts in the annual cycle where the correct choice is really a harsh one, and you got to select the right one or the consequences are steep. What I'm thinking about is arrival to breeding areas in West Greenland. So we see massive flexibility in when birds migrate from wintering areas to staging areas. We see almost no flexibility in when they arrive on breeding areas. So 3-week advancement over the last 30 years in departure from wintering areas, a 1-day change in the arrival to breeding areas. So to me, that suggests that a few individuals tried it. They're not around anymore.
Yeah.
Or if they are around, they barely made it on the snowpack in West Greenland, and they didn't do that again. So there's quite a bit of evidence for only specific parts in the year as being really important to make the right decision. And so all this factors too in how you might more properly parameterize this kind of ecological memory model.
Thank you. It's very interesting.
What else in person?
Yeah. So I was thinking about, I don't know. I've got it very loud.
Well, it's so the people on Zoom can hear you.
He's thinking about, you've got those 7 separate subpopulations of white throats. Are they still ongoing?
In terms of devices alive, yeah, they're all still going.
Doing the whatever.
We're still collecting data, yeah.
A treatment of creating a super free, just massively feeding these birds. Play themselves on the breeding ground.
You mean a wild birds you're talking?
Yeah, like, for example, I did a 50, got a food drop Carrot Lake on Tuesday, right? Let's throw 200 bags of corn in there or something and people.
Yeah,
You see what I mean?
yeah, I see that, but yeah, it would be hard. You mean, so you're gonna invoke some response?
Just gonna invoke a response.
Yeah, yeah,
Well, no, because, okay, they're not starving. They chose not to breed.
yeah. That would be so, yeah, that would,
Then it's-
that,
Not food.
yeah, that would be phenomenal, Steve, if you could do it at the scale to measure variation. Like, it's just, it's so hard to do this at a meaningful scale. But on a micro level, you could probably do this like in captivity even, with wild birds, which we sometimes
I mean,
do.
If we were gonna do something in captivity, I'd probably do a common garden experiment on eggs. And.
Yeah, you could do it through life.
They speed up.
Yeah.
Speed up, right? You just switch them around.
That would be the way to do it, I
Everything
think.
from 7 colonies
Yeah,
To see that, has that been done yet?
no, that hasn't been
would
done.
be-
That would be cool, yeah,
Yeah.
for sure. Yeah, so it's somewhere in between. You'd start with those experiments and
But
maybe you
just
could
so
do it
that
locally
we're-
after that.
Okay, so rather than looking for that in their food productivity, let's just push it so that they don't have that feedback. And are we looking at the right parameter?
Yeah, no, that would be great. I think the scale is hard, but in captivity, you could begin to get at it.
We got to start somewhere.
Yeah,
So. can tell it some-
yeah, I think you could. I think, yeah, definitely. A motivated person could start to assemble the birds and start to compare this stuff. Yeah, I've got there are a couple things here. Let's see, Andrew, I'm just reading this comment here. White nose syndrome in bats, interesting common feature. Fertilization. Yeah, that's great, Andrew. Really appreciate that. Do you have a specific question that is associated with your comment there? No? Okay. Interesting point. Maybe email, not at the moment. Okay, email if you do. I think we can certainly exchange some ideas. It sounds like you're interested in a lot of similar questions there. Ryan, go ahead.
Hey, Mitch, great talk. I wish I could be there and buy you a beer after, but really interesting what you've talked about. I wonder if you have any examples where hunters or other people on the land have informed your hypotheses or even given you some light bulb moments around some of this research.
Sorry, Ryan, I missed the first part of that. Could you ask again?
Sorry, just wondering if through all of this work in talking with hunters and other people on the land, if there's any times where that has really informed hypotheses or given you some light bulb moments around this sort of large scale research.
Are you thinking about the individual or the population level or both?
But anything, just where obviously this is all very quantitative science, but at the same time, I've had experiences where I thought I had things figured out, and then somebody had a point or a question, and then I went, "Oh yes, I hadn't thought about that." And these lay people just think about things differently and give me lots of ideas.
Oh yeah, for sure, lots of moments like that. So 2 really quick examples where we work in Iceland with the green and the white fronts. Like the farmers, they know exactly the patterns of these birds. And so they'll tell you they feed on this at this time of season and they feed on that at this time of season. And we can do some neat spatial analyses to suggest where they go and why they go there. But the farmer can tell you that in 10 minutes over coffee. And and I don't mean to say we're not doing good stuff. I mean to say the ideas that these folks have that they think are so simplistic are actually quite complicated ecologically. So that's always very exciting. Here we're going to be in Iceland meeting with some of these folks in the next few weeks. And so I assume more of those light bulbs will be coming. So yeah, I mean, there's a super energetic cultural component too, that it's fun to interact with these folks who have these birds in their backyard and don't think much of it. And yet these birds are connecting cultures and landscapes. So there's a lot of excitement in those conversations, at least from my perspective. In the case of the pintails, like at a larger scale, there are a lot of, I know you know a lot of this stuff. There are a lot of challenges working with producers with a conservation hope. And yet you meet with these folks, they're really good people. And they have a good feeling for where these birds are nesting, where the areas are not so profitable on their farms. And so I feel like the light bulb moments there are how do we work in a more meaningful way, have quality conversations, starting from a positive place to better understand pintail conservation and more broadly, animal conservation in a seriously changing landscape. I get they want to make money, they have a livelihood, but also a lot of them have a sense of keeping the land in a particular level of quality for future generations. And so the aha moments there are having quality conversation about how do we move this massive challenge forward in a meaningful way. And we can do that among our partnerships, but including these producers in some of those conversations has been really inspiring, hearing about their motivations for their families and future generations. And so that's always quite enlightening to me. Any other questions or comments from the group? Okay, thanks everyone. Appreciate it.
Awesome. Thanks everyone for joining.
Good job, Mitch.
Thanks, Phil.